because they're so small we can't see them with our light microscope. So one resource are the photomicrographs that Professor Holland has downloaded and printed for us. So here's a cartoon of the normal cellular prion, the disease-causing prion. Here's an example of the spongiform encephalopathy. The disease-causing prions causes a stained thin section through the brain of somebody that died of a prion disease. So these are resources, and be aware, you guys, if you see diagrams up here or photos up here, they could appear on the lab practical. Okay, so don't ignore that. So prions. The influenza virus that's mentioned in Cast of Characters, we don't have slides, but again, we have a photomicrograph, an electron micrograph of influenza viruses here for you. This next one is a photomicrograph of mycoplasma, which is a very small bacterium, which lacks a cell wall, and amoebavirus, one of the recently discovered huge viruses, showing that some viruses may be as big as some of the small bacteria. Okay, so some resources here. So those are the photos at the side that we're discussing there. Also, fantastic resource, you guys. Last semester, 2 of your colleagues made these dynamite Cast of Characters posters. There's one on the pinboard there and another in the back. They made those posters based on the Cast of Characters lab. And you can see they organized all the information that that's so helpful in studying for the lab practical. So those are dynamite resources. In addition, if you use the index of your textbook, use the index, look for the name of the organism, the character, and often your textbook will have some dynamite photos of many of the Cast of Characters. Your photo atlas as well, although I've been often disappointed in the photo atlas. It doesn't have all the organisms we look at, but certainly the photo atlas is helpful. Another technique some of your colleagues have used in the past is they'll do a Google image search. Like for example, they'll go to the search box, they'll type in Spirogyra, one of your Cast of Characters, and they'll type in Spirogyra image, and that will bring up all the photomicrographs that have been posted online of these organisms. And what some of your colleagues have done in the past is that they'll print those photos, cut them out, attach them to index cards, and then write all the important information on the back of the card, and they use them as flashcards. They'll take them shopping with them, so if they're standing in line, they'll just go through their flashcards. It really helps just using those little, 5-10 minutes here and there. If you got some flashcards in your pocket, just pull them out, and in no time. You can show them to your dog or cat or to your kids. They will all listen to you as you're talking about all the wonderful Cast of Characters here. Okay. Yeah, and this is just to remind you guys, with regard, and I totally understand, new course, you guys are trying to figure it out. So yesterday your colleagues were saying, well, what should we expect on the lab practical? So expect you could see all the Cast of Characters on the lab practical. With those that we have microscope slides, what we do is we cover up the name of the organism. So we'll cover up the name of the organism, we'll put it on the microscope, we'll have you look at the organism, and usually, of course, the first thing you need to know is, what is it? So you'll need to be able to identify all the Cast of Characters, from the slides or the photos that are provided. And then once you know what it is, who it is, then we can ask you all kinds of questions. And most of the questions are based either on the questions that are asked in the lab manual itself, or they're questions that would come from this table that you fill in. For example, classification. For example, is this microbe considered beneficial or a pathogen, disease-causing? If there are specific structures that perhaps we talk about, we might ask you to identify a structure. Okay, so the hard thing that for a lot of people is just this concept of sitting down at a microscope, looking at a specimen, and knowing who it is. Okay. And again, folks, this is where I think drawing is really helpful. It helps you to identify unique shapes of the cells. Size is really important. Knowing the size tells you a lot about who or what this organism could be. All right, now, just to kind of give you the overview of the Cast of Characters, what I did is I tried to put our roadmap up here on the board, just to help help remind me where the specimens are located. So if we start with the cast of characters that are listed in your lab manual, you'll see the list starts, in my version of the lab manual, it starts on page 57. Part 1 is titled Acellular and Prokaryotic Microbes. Okay, so they give you some study hints there. We turn the page, and we start with prions and influenza virus. Again, we don't have slides for you. These guys are too small to be seen. So we would refer you to the photomicrographs and the diagrams on the side. Also listed are pages in your preferred textbook, and in some cases a photo atlas that you can use as a resource. Okay, so those are our two acellular organisms. Then if we move on down, we run into Bacillus anthracis, Neisseria gonorrhea, and Anabaina. These are cellular organisms. So cellular, they're prokaryotes. They belong to domain bacteria. Now, Neisseria gonorrhea is a bacterium that causes sexually transmitted infections. They're very small. You really need an oil immersion lens to see them clearly. So since I'm requesting you not use that lens till next week, I set up a demoscope over on the side. And oh, these samples are great, you guys. What happens in gonorrheal infections, especially in men, is when the bacteria colonize the urethra, it causes a real strong inflammatory response, traction of a lot of white blood cells, and so often men will just develop a urethral discharge. It's just like pus, and pus has lots of white blood cells in it. So these slides have been made, these are smears of that urethral discharge from folks suffering from gonorrhea, and they are beautiful. I mean, you're the poor person, right? But they're beautiful smears. And what you'll see when you look in the demoscope here is you're going to see lots and lots of neutrophils, white blood cells. Those are the first responders. Their job is to arrive quickly on the scene when we have microbial infection and try to phagocytize and destroy the invading pathogens. And what's so beautiful is that you're going to see all these human neutrophils, and we're going to get the pointer on a neutrophil that has successfully phagocytized some Neisseria gonorrhea. So what you'll see in these neutrophils, they have a multi-lobe nucleus. Okay, so I'll just draw a little neutrophil here. They're going to have a multi-lobe nucleus. And what we'll try to do is put the pointer on one that has successfully phagocytized the Neisseria gonorrhea. And the Neisseria gonorrhea, they're little cocci, little ball-shaped organisms, and they're often in pairs. Okay, so this would be the neutrophil with a multi-lobe nucleus, and then inside you're going to be looking for the bacteria. Okay, so that's the demoscope on the side. The Bacillus anthracis, this is the bacterium that causes the disease anthrax. They form long chains, long chains. They are rod-shaped or sausage-shaped cells. When they divide, they often stick together, forming these long chains. And even at 40x, you can make out these long chains. They almost look like little worms or little hairs. Okay, so that's what you'll be looking for in your micro B box. We go down here, Bacillus anthracis. Where are you going to find the Bacillus anthracis slide? It tells you right in your lab manual. I just want to make sure, because a lot of people can't figure this out. The micro B box, right? It'll be in your micro B box, and which slide will it be?
Number 1.
Slide 1, right? Okay, so the lab manual will tell you which micro box, which slide. Good. Anabaina. Anabaina is a cyanobacterium, very similar to nostoc, carries out oxygenic photosynthesis, carries out nitrogen fixation. Okay, I couldn't find the prepared slides, so we have something even better.